This podcast will provide a brief overview of the lymphatic vascular system. So we'll be talking about lymphatic vessels, lymph fluid, and lymph circulation. Now, the major lymphoid organs, which we're going to get to in subsequent podcasts, include the tonsils, lymph nodes, the thymus, Peyer's patches, and gut-associated lymphatic tissue, the spleen, appendix, and bone marrow. I just want to highlight for you right now, the thymus and the bone marrow are primary lymphoid organs. Lymphatic vessels would be initial lymphatic capillaries, collecting lymphatic vessels, and eventually the right lymphatic duct and the thoracic duct. Just to put this cartoon in a little bit of an overall perspective, we know that immunocompetent T and B cells will seed the lymphoid tissues, or these lymphoid organs, and as immunocompetent cells are becoming activated, they're going to respond to an immunologic challenge. So we know the T cells will originate in the bone marrow and migrate to the thymus, where they'll become immunologically competent, and B cells are either going to remain in the bone marrow to mature and become competent, or they'll mature in the other peripheral lymphoid tissues. Mature and immunocompetent cells circulate among the various lymphoid organs using either the blood or the lymphatic vessels. So I want to talk in this podcast a little bit about the lymphatic vessels and their circulation. Lymphatic vessels generally parallel the blood vessels, but there are some exceptions. So, for example, there are no lymphatic vessels found in the central nervous system. There are no lymphatic vessels found in cartilage, and as you know, there are no blood vessels found in cartilage either. There are no lymphatic vessels found in the bones and in teeth. There are no lymphatics in the placenta, and there are no lymphatics in the parenchyma of the thymus. There may be lymphatic vessels in the connective tissue associated with the thymus, but not in the parenchymal tissue of the thymus. And by parenchymal tissue, we mean the functioning tissue of an organ. So there are no lymphatics in the parenchyma of the thymus. Lymphatic capillaries originate as blind-ended vessels. They are composed of a single layer of endothelial cells, and unlike blood capillaries, they're not associated with pericytes. They're not fenestrated. They don't have tight junctions, and there's virtually no or very little basal lamina. They have a much higher permeability than blood capillaries, and they can branch and anastomose freely. And as we'll see in the laboratory, when you're trying to identify lymphatic capillaries, oftentimes you're looking for what appear to be almost just a hole in the tissue section. You almost don't see a structure. There's such a thin wall to them. Lymph is considered an ultrafiltrate of plasma, so it's essentially water, electrolytes, and perhaps a small amount of protein, maybe 2 to 5% protein. Lymphatic circulation is unidirectional. It flows from the tissues back to the heart, so we would appreciate that lymphatic circulation begins in the lymphatic capillaries out in the tissues of the body. These lymphatic capillaries will get larger and merge, coalescing into lymphatic vessels. These will get larger and larger, merging eventually into the thoracic duct and the right lymphatic duct, which is probably one of the largest of the lymphatic vessels, and these become confluent with the jugular and subclavian veins. And you probably have an appreciation for that from your study of human gross anatomy. Lymphatic vessels have structures that are similar to veins. At least the larger lymphatic vessels have structures similar to veins, but the walls are thinner. There's no distinction between the tunica, intima, media, and adventitia. They have relatively large lumens in relation to the wall thickness, and they may have valves to ensure unidirectional flow of lymph fluid. So here is a very large distended lymph lymphatic vessel showing the valves in the lymphatic vessel. And here again, a relatively large lymphatic vessel, very irregular, very distended, showing valves. Here's a corresponding artery, a corresponding vein, and for the same bore size for this artery and vein, look how much larger and more distended the lumen of the lymphatic vessel is. Here's an example of a very large distended lymphatic capillary in the GI tract, in the villi in the GI tract. This is called a lacteal. Lacteals are found in the lamina propria in the small intestine, and so here is an enfolding of the small intestine. Here are individual villi, and here would be the spaces of these lacteals. So this is just shown in a higher magnification here. This is the well-distended lymphatic capillary. Lacteals transport chylomicrons, lipids basically, to the liver. Chylomicrons are synthesized by the intestinal epithelial cells, and the chylomicrons are exocytosed into the lacteal, where they're transported back to the liver. So the principal functions of the lymphatic vascular system, from the point of view of a general circulatory function, the lymphatic vascular system returns lymph fluid and plasma proteins to the blood and transports chylomicrons to the liver from the small intestine. From the point of view of a lymphoid function, the lymphatic vascular system adds antibodies that are formed by B cells and lymph nodes to the blood and to the lymph fluid, and the lymphatic vascular system will carry lymphocytes, primarily B cells, to various lymph nodes. As you know, lymph nodes are found along the course of lymphatic vessels. We'll talk about lymph nodes in more detail in a later podcast, but lymph nodes can function as inline filters for lymph fluid. So in the cartoon, you can see these lymph nodes in chains to each other and serving inline filter, and because all lymph fluid is filtered by at least one lymph node before the lymph fluid returns to the blood circulatory system, the lymph node has the ability to add antibodies into the lymph fluid. Now, importantly, these lymph nodes, as they're functioning as a filter for microorganisms or tumor cells, that can cause a problem because it is possible for infections to spread along the lymphatic system from one area of the body to another. You know from your clinical exposure, or you will know from your clinical exposure, that this has significance because you get systemic infections and you can get metastasis of tumor cells very easily through the lymphoid system, through the lymph nodes. And lymph nodes, if they are harboring an infection, can actually swell, get hard. They can actually be palpatable on a physical exam.